All right, everyone, we're going to get started. Thank you for everyone who's joining us online and in person today. My name is Karen Jacobs. I'm an imaging scientist here in the IDM here at the University of Cape Town. And I'm very happy to welcome you to the first in what I think is going to be quite an exciting new seminar series that we're co-hosting with Lysis. With the focus on really wanting to highlight the actual applications of modern microscopy in disease and other research areas are really of interest in Africa. And we really want to focus on the work being done by African scientists and work being done in an African context. But with that said, I think today's first session is we've kind of designed it as a bit of a taster menu that everyone can get access to or get a taste of research being done in a few different topics, specifically what we refer to as the big three, HIV, TB, and malaria, and then upcoming sessions that we'll mention again at the end of today's session will focus on one topic at a time, again with multiple speakers. I think that's enough of a welcome from you for the session. So I'd like to welcome up our first speaker for today. And he's actually, well, it's a team of speakers presenting in person today. Prof. Digby Warner, who is the director here at the Institute of Infectious Disease and Molecular Medicine, but today speaking as the head of the Mycobacteria Research Group, focusing on the work that they do in understanding the biology of mycobacteria. So Digby, over to you.
Thanks, Karen, for the introduction, and welcome to everyone in the venue, and also to everyone online. I hope you can hear me clearly. It's a great honor to get to lead off this series, which, as Karen mentioned, has been co-organized by the Africa Microscopy Initiative and Lysis, and is being hosted here within the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town. I'm going to give a brief introduction to the team of people who actually do the work and are the real experts in microscopy, and allow them then to speak in a little bit more detail about how the use of imaging has transformed our approach to research of one of the world's leading pathogen and causes of death by an infectious agent. So in advance of the session, Karen asked us to really focus on answering 5 questions: where we're from, what our key research areas of interest are, what imaging instruments or technologies we use in our research, why we chose those instruments, and how the imaging has actually helped us gain knowledge and advance our research. I'm only going to address the first 2 of those because, as I mentioned, I'm going to hand over to the real experts in the team who really are leading the last 3 components. But by way of introduction, we are based here at the University of Cape Town in the IDM. As already mentioned, we're part of a large research group which focuses on Mycobacterium tuberculosis as the causative agent of tuberculosis. TB has just regained its status as globally the leading cause of death owing to an infectious disease. And here in South Africa, which is where Cape Town is located, we sit and have done for almost 2 decades at the nexus of 3 coalescing epidemics. That's drug-susceptible TB, multi-drug resistant TB, and TB HIV. And a tuberculosis prevalence survey conducted countrywide just before COVID, so in about 2018, 2019, confirmed there was the statistics which most of us were familiar with already, which is that our prevalence sits at close to 1% of the population, and that very importantly, a very significant amount of TB disease goes unreported and undetected in our communities. So within our research group, we focused on 3 broad areas. Firstly, that we need a pipeline of new, better TB drugs, and how can the basic science inform these approaches. We need to protect against TB drug resistance. And we also need to understand why TB rates are so high. And in putting together these brief slides, it struck me that for all three of those questions, we now use imaging tools as part of our essential research arsenal. So as I mentioned, I'm not going to look at the last three questions, but what I will say for those who are interested in getting into microscopy, that when I looked back at previous presentations, as recently as 2014, I was still giving talks where the kind of images I would show are those which are projected on your screens now, where the best way of looking at Mycobacterium tuberculosis was observing colonies on plates or looking at optical density during liquid growth. That was the sort of the best resolution that we were using in order to understand the biology of this organism. Over the last decade, since we've now switched in a big way. And so some recent work coming out of the group, which I think I'm hoping will be discussed in future installations of the series, has started to use a combination of advanced microbial genetics and imaging tools in order to understand fundamental metabolic adaptations of mycobacteria, and also to give us insight into the molecular machines which might be targeted for new anti-TB drugs. But that's where I'm going to lead it. I should also say that the work that will be presented that has been presented by me and will be presented by others is funded by many people who we must acknowledge, but owing to the demands of time, we won't spend a lot of time on that. So I'm going to hand over to Dr. Ryan Dinkley from the group.
Thanks, Digby. Thank you, everyone, for attending and for this opportunity to present my research. It's always great to have a captive audience who has to listen to you. And so my role in the MMRU is a little bit different. I'm not a hardcore microbiologist. My research interest is really focusing on the aerobiology of Mycobacterium tuberculosis. What is that? What I study is everything associated with the takeoff, flight, and landing of mycobacteria from infected individuals to new hosts, with specific interest on Mycobacterium tuberculosis release or aerosolization. Briefly, why is it something that we want to study? When you look at the natural history of tuberculosis, you very quickly see that transmission is an essential bottleneck. If we could eradicate transmission overnight, it would no longer be a problem. That being said, we've been studying transmission for almost 100 years now, if not slightly more, and we're still not really closer to that being a reality. Presently, there are only a few techniques that are capable of enumerating by aerosol release of Mycobacterium tuberculosis, and they typically rely on either the detection of DNA or RNA or culturing the organisms. These are, although they're great, they're problematic in that we don't get any insights into the physiological characteristics of the organism which enable transmission. To this end, we've designed and developed a novel microscopy-based approach to detect bacteria that have been released from TB patients. We use a fairly simple instrument. It's a Zeiss X-ray Observer 7. It's really robust and it does everything we need. We have wide field fluorescence as our illumination source. We specifically use LEDs. For our analysis, we're not doing super advanced hardcore microbiology. Rather, our main primary output is how many bacteria are people releasing. And so we have a manual bacterial enumeration that happens at the scope. We're working towards automating this process for several reasons, but given the challenging nature of the samples, it's proven challenging. We also use Fiji and Microbe J for quantifying bacterial morphology and specifically the staining distribution along the cell, as these are informative parameters for underlying cell physiology. Why we use this setup? Bi-aerosol samples are typically fairly low quality for high resolution or super resolution techniques. So in order to get significantly better resolution, we'd need to sort of clean up our samples more, which is always challenging with a clinical sample. But wide field microscopy also allows for a lower light dose and just generally is easier for scanning broadly large samples. And so you can spend a lot of time looking down the eyepieces on the system, and it's really well suited to this application. In microbiology, you'd be surprised how great the images can be with wide field. you can get away without, especially confocal for most applications, although we do use a LARA if we need super resolution imaging. The results of this, we've been able to detect and quantify the aerosolization of Mycobacterium tuberculosis from a variety of participants, including TB patients, over the last several years, which have led to several publications and contributions to the field. First and foremost, we saw that we could identify aerosol phenotypes. We don't yet know the significance of these. There seems to be quite a few, but bacteria aren't all aerosolized in the same state, which is intuitive, but we're starting to show that. We also detected bacteria in small clumps and clusters, which has potentially important implications for downstream infection. More recently, we demonstrated that these bacteria could be aerosolized independently of coughing, and that actually the amount of times you perform a respiratory maneuver per day is really the primary driver of aerosolization. So, for example, a person can cough 350 times a day, maybe 500 towards the upper end, whereas you breathe 24,000 times a day. So by sheer magnitude of breathing, you ultimately will breathe out more bacteria just breathing. And lastly, which was published, I think, this week in PNAS, we demonstrated that MTB are released from all TB clinic attendees, irrespective of diagnosis. And even more surprisingly, these people that are attending the clinic diagnosed as not having TB and aerosolizing the bacteria clear their bioaerosol sample at an equivalent rate to TB patients diagnosed as having TB and receiving chemotherapy, which suggests that the clearance of the sample is primarily immune-driven. So as you can see with microscopy, we've had some novel insights into the aerobiology of MTB, and we're looking forward to the discoveries that are still to come. Again, I'd like to acknowledge the following and then hand over to Joseph.
Hello, everyone. Thank you for having me here today. So I'm a PhD student based in Professor Digby Warner's group. And I'm going to share some of my work, my PhD work. And so we are investigating the use of phage, mycobacteriophage, for the purpose of lysing mycobacteria, planning to achieve two objectives. Number one, for DNA extraction. And number two, to see if we can be able to use this phage as an adjuvant in killing mycobacteria. So we use this system whereby we are combining fluorescence microscopy and cell ASIC, which allows us to do live cell imaging over time. And maybe just to give a brief touch on the cell ASIC plate, you can see where we load our different conditions. And we are able to open channels, and we can be able to image whatever is happening to the bacilli under different treatments. So by this, we use fluorescence. For example, we can use we can be able to investigate permeation of antibiotics by measuring the fluorescence. And for some of my results, we have demonstrated that the mycobacteriophage actually lyses the mycobacteria. And what I'm showing here is the viability, whereby on the left side you can see that the viability is lost, and we are achieving close to 100% killing of mycobacteria. And in the middle panel, I'm showing the efficiency in DNA extraction, whereby after lysis, we try to recover this DNA and quantify it using ddPCR. So we have done this in M-SMEL-MATIS and also in MTB. And what we can see here is that a comparison of the standard methods of DNA extraction versus the phage method, we are able to achieve close to 100% DNA recovery, especially in low density samples. And for my second results, where we are using the mycobacteriophage to compromise the mycobacteria cell wall, and we measure the permeation of antibiotics. And what we can see here is that there is a dose response. If we use low amount of phage and high amount of phage, you can see that the permeation is highly achieved. So this is the fluorescence intensity quantified, and purpose of this, we use PG and image J. So thank you very much.
Digby, Brian, and Joseph, thank you all very much for that great taster talk. That was fascinating. I want to open up, we have a few minutes open up to the audience if there are any questions. We'll start with in person, and then I'll ask if there are any questions online for the speakers we've had so far. Don't be shy. Okay, it looks like we don't have any questions in the room. No questions online. Okay, and it looks like oh wait, there's a question from.
What is the magnification that's suggested to look at TB?
Just repeat the question online.
So the question was, what is the suggested magnification to look at TB? I think it depends on your application. So for us, we've been using 100 times objective for the aerobiology work, at least, with a numerical aperture of 1.4. So we get sufficient resolution with that to get decent quality images, although the contrast is low because of the background. In terms of magnification itself, that's helpful when you're scanning manually to scan a bit of a smaller area, although it does take a bit longer when you're looking through the nanowells. Although we are moving towards a 63 times objective simply because it has a bigger field of view. So in my experience working on the ELIRA, for example, in the BSL-3, a bigger field of view means fewer images to get the same number of bacteria. So it increases the throughput of your process. So as long as your resolution is sufficient, 63 times or 100 times are fine.
Brian, thank you so much. Any other questions? Yes, in the back.
I'm not sure if you can hear me, but I think it's a question for Diane as well.
Yeah.
So just about the aerosolization, breathing and coughing of those diagnosed with and those who aren't diagnosed. I'm just wondering about the viability. Were you able to actually assess the viability of what?
So the probe that we use is metabolically incorporated into the micromembrane. Sorry, I didn't have a lot of time to go through it. And so it is inherently a viability marker. If cells are fluorescing, they're alive. It doesn't exclude the fact there might be viable organisms that we are not detecting, who are not taking up the stain for whatever reason. But yes, it is suggestive that they're alive. We found in individuals with GeneX with positive confirmed TB that at the end of treatment, so 6 months after being treated with antibiotics, about 20% of them were very low positive for aerosol bacteria still, which is consistent with previous work looking at bronchoalveolar lavages and mRNA actually. So it is a consistent ballpark estimate with what we would expect to still be producing MTB at the end of treatment.
Anyone else? No other questions. Okay. Thank you once again to our in-person speakers. I think that was a great way. Oh, there's a question online. Is the mycobacteriophage adjuvants are successful in the lab, is the next step preclinical animal experiments and then phase 1 trials? I think that's for you, Joseph. If you want to try answer it.
Yeah, so what we are doing, okay, we've not gone to the extent of this is basically experimental work.
The question's here if you want to see it.
Okay. So, yeah, we definitely go into animal model studies. So I hope that answers the question.
Cool, simple answer. Thank you. We're going to move on to remote presentations now. Our speaker from Ghana is not yet online, but we have 2 speakers joining us from ARI in Durban, Marantha and Trevor. I'm going to invite you to start sharing your screen while I start introducing you. I hope you're still online.